Hello, dear learners, students, and professionals. Welcome to the very informative video on stages of pharmaceutical product development. In this video, we will study the stages involved in the formulation development or product development. The stages are very much important for development of generic formulation and as well as the hybrid formulation or the innovator formulation. So the stages will be similar and the stages will have much more impact on the product development and outcome of the product development. So after watching this video, you will be able to understand different stages and the studies involved in the different stages of pharmaceutical product development. Mainly, I have divided the stages into 6 parts, and these are literature search and pre-formulation studies. Second is prototype development for formula and process. Third is scale up and pilot studies. Fourth is demonstration of bioavailability and bioequivalence. Fifth is commercial manufacturing. We may call it as routine production, and the life cycle approach, which involve the sixth stage, that is changes to the approved formulation. These changes may be to the composition, process, and site. So these are the 6 stages of pharmaceutical product development. Also, this is the life cycle of the pharmaceutical product. Coming to the first, literature search. See, literature search is a never ending stage. At all the stages, you will need literature backup and literature survey. Then pre-formulation studies. Before starting any formulation development and prototype development, we have to understand the API and the excipients for any of the formulation. That's why literature search and pre-formulation studies are done. See, in literature search, the specific and relative information from available reliable literature to be taken into consideration. Literature is very vast topic, and on the same point or on the same required information, much more literature is available. But whatever the literature we use for our understanding, that should be reliable. Search approved formulation literature. Also, we have to consider the formulation literature for those products which are already approved. So here I have mentioned search approved formulation literature. Then literature from the regulatory websites, published articles and the patents. Always have to search the reliable literature. Otherwise, if literature is not proper, our direction may change. That's why the literature should be very specific, reliable, and it should be from reliable source. As I have mentioned, literature search is never ending stage, so it is required and is ongoing throughout the product life cycle. It is not a stage like you have done literature and completed it. No. Whenever you will face problems, that time come back to literature or come back to pre-formulation studies, search it, get the answer and go ahead for the next stage. Then coming to pre-formulation studies. In this video, I have included mainly the pre-formulation studies. In pre-formulation studies, we have to understand the physical, chemical, biological and the mechanical properties of the drug substance. And all these properties are related to the product. If you consider a product of tablet or capsule or suspension or solution, all these information are required to be available with you before starting the formulation development, so that if you face any of the problem in the later stages, you can come back to pre-formulation studies results, get some idea, and then you can relate those things to the later stages. Out of this, in this slide, I have mentioned physical properties of the drug substance. So what are the physical properties of drug substance? That is particle size, particle shape, thermal transitions, solid state form, that is crystalline or amorphous or polymer, then density, powder flow, solubility, hygroscopicity, chirality and stereoisomerism, and moisture content or water content or loss on drying. So preformulation is vast and a single API required to be studied in depth because these properties affect your formulation. These API properties will affect your bioequivalence. These properties will affect your process. These properties will affect your stability and so on. So preformulation studies are much more important. Particle size distribution involves 3-tier particle size specification to be given to the API by sieving method or diffraction techniques. Particle shape. Microscopic studies can give you the idea for particle shape. Then thermal transition for API by hot stage microscopy to visually examine all kinds of thermal transition when the sample is heated or cooled. That means how your API behaves under the influence of heat. Then solid state form. Is your API crystalline or amorphous? Or is it shows polymorphism? Is it solvate, hydrate, pseudopolymer? So this study can be done by PXRD and DSC. Then bulk and tap density required to be determined. Then powder flow is required and is done by Carr's index, Hausner ratio, angle of repose, and studying the compressibility characteristics. Then coming to the solubility. Solubility itself is a very vast topic, and solubility involves mainly the intrinsic solubility. Then BCS solubility and sink condition. Then saturation solubility, solubility in the solvents, and solubility in the biorelevant media. Then solubility as a function of pH, that is pH solubility profiling. So all these solubility characteristics are very much important for your dissolution and bioequivalence or bioavailability. So these physical and chemical properties may affect your biological properties. Then hygroscopicity. It is required to determine the nature to absorb moisture from surroundings. Then coming to chirality and stereoisomers, these are required to be checked and determined for the product under development. Moisture content, water content or loss on drying. So is there any moisture content, water content or loss on drying for the API that is required to be determined? Because these are the API CMAs, that is critical material attributes, which affects your formulation. Then coming to the chemical properties of drug substance, it involves pKa, that is dissociation constant. It determines your API's acidic or basic behavior. Then pH of API. Generally pH of API is done to determine the nature of the API, and generally 1 or 2% solution in water is prepared and the pH is checked. Then chemical stability. So chemical stability involves stability in pack conditions, stability of the molecule in dissolution media, force degradation data from API vendor. Force degradation is also available in the DMF that is required to be studied. Also, many times the API is studied for force degradation depending on the product requirement. Then photosensitivity and photostability. Melting point and glass transition temperature. These are required to be studied by DSC study. Generally, whenever the heat is involved into the manufacturing process, that time melting point and Tg is considered. Then impurity profile. This is a very much important topic for any product and any market. Nowadays impurity profiling is very much important, which involves residual solvents, process impurities, degradation impurities, isomeric impurities, nitrosamine impurities and the elemental impurities. Nowadays, recently many guidelines are being generated for nitrosamine. So this also is required to be understood. Then coming to the biological properties of the drug substance. These biological properties mainly affect the bioavailability and bioequivalence. So these are partition coefficient, log P, we call it as log P, octanol water partition coefficient. It gives idea about the nature of molecule, whether it is hydrophilic or lipophilic, and what is the ratio of this hydrophilic lipophilic nature. Then permeability study. CACO 2 cell permeability studies are done, and based on that, whether the molecule is having high, low or moderate permeability is determined. And BCS classification of the molecule is required to be understood. There are 4 BCS classes: class 1, class 2, class 3 and class 4. High solubility, high permeability belongs to class 1. Then class 2 involves low solubility and high permeability. Then class 3 involves high solubility and low permeability. BCS class 4 involves low solubility and low permeability. Now, in some of the literature, you will find the BCS class 2A and 2B. So this is a type, 2 types of BCS class 2 molecules based on their pK. You can get literature on that. You study that literature and you will come to know the sub classification for BCS class 2. Then sub molecules fall under the class of 2 and 4, because as a solubility they are low soluble, and as permeability they are moderately permeable. So this can be referred as the molecules which fall in between 2 and 4. So borderline BCS also you can say, and sometimes BCS is not clearly understood based on the drug molecule. Now this risk assessment of the drug substance for formulation based on its attributes. So all these physical, chemical, biological and mechanical properties are required to be studied, and based on this risk assessment is done for the formulation. Now we will study the mechanical properties. This topic may be new to the many of the professional students and learners. See, mechanical properties are very much important in the actual working or actual development. How the API or drug substance behaves under stress condition, whether it is plastic or elastic, what is the brittleness, is it ductile or not, what is the brittle fracture index of the API, and based on these mechanical properties we can understand the product development requirement. So elasticity, plasticity, hardness, brittleness, tensile strength and fracture toughness are understood from the stress and strain studies. The molecule is given a stress and its behavior under that stress is studied. The response of material to stress will be reversible or it may be irreversible. If it is reversible, it will be elastic deformation. Like you give stress and it will bounce back. That is elastic deformation. Like a elastic. Or the stress impact may be irreversible. the irreversible impact may be brittle or ductile, and we want the plastic deformation or ductile deformation many times when you consider the tablet formulation. More plastic nature of crystals or API molecules will give good tableting property. Why? Because the stress will be taken into the molecule and plastic deformation will be there. Brittleness, ductile nature evaluation of API and crystals. This is very much important when you are dealing with a high dose formulations like 1000 mg dose and tablet weight is 1200 or 1100 mg. So that time all these properties will play a very critical role, and a formulation scientist should understand these things. Change of crystal form or melting, degradation due to mechanical stress. This may happen for some of the very delicate molecules. If you give mechanical stress, the API may get melt or may generate impurity by degradation or by mechanical force like in milling and compression. Then deformation characteristics, plastic and elastic deformation. So this we will try to understand from brittle fracture index, and one another terminology is there, that is ductile fracture index. See, ductility is a very important characteristics of metals, and brittle fracture you can understand from glass. If you break the glass, it will break into number of particles, that is brittle material. If you apply stress on metal, it will not break, but it will be deformed. That nature is called as ductile. So BFI and DFI are two terminologies. Lot of literature is available on this. You can search and get more idea. BFI has been used as a measure of plastoelasticity of the pharmaceutical powders. See, if your material is not having a good plastic deformation properties, then you may face capping and lamination or less hardness in the tablet formulation. The BFI value has a range of 0 to 1, and 0 means no fracture tendency and 1 means maximal fracture tendency. The tablet samples with BFI values less than or equal to 0.5 displayed a high fracture incidence during the actual tableting. Based on the BFI nature, we have to understand the formulation requirement, and binder are required for the drug substances which have a very little deformation tendency and which have a more brittleness tendency. Then after preformulation studies, one more interesting topic is there, that is drug excipient compatibility studies. We call it as DEC studies. DEC studies are done for understanding the compatibility of API with the excipients, and based on that excipients are chosen and used in the formulation. There is no specific guideline for drug excipient compatibility studies. That's why different people have different understanding, but the motto is same for all the scientists, that is to have a compatible excipient in the formulation. Then it comes to selection of suitable formulation as per the dose and therapeutics. If you are making a 505B2 or if you are making a novel formulation, that time the pre-formulation studies or drug excipient compatibility studies are determined based on the dose and therapeutic category of the API and the formulation. If you select tablet, capsule, granule pellets and many more, if it is oral formulation, many times different formulations are made for the same API, and then the best formulation is chosen based on the different properties like dissolution, stability, bioavailability and all. In drug excipient compatibility studies, we make the mixture of the API and excipient, and we keep it for some time, maybe 1 month, 2 months at different conditions to understand the behavior of the API and compatibility of the API with the excipients. In this, mainly the physical observations are done. Then degradation of API in contact with the excipients and solvent is done. Impurity profiling is done. The drug is studied for impurity generation. Then also in this DEC, we have to study the stabilizing effect of excipients or degradation impact of the excipient, considering the storage condition and excipient ratios. This study will give the information on degradation pathways and potential processing conditions for the drug product manufacture. If your API is unstable with solvents, let's say water or let's say acetone, then you can understand from the DEC studies that the solvents like acetone and API should not be come into contact, or water cannot be used. So based on these DEC studies, lot of information can be gathered, and based on these preformulation studies, the prototype development can be done in a best way. In summary, in this video we have understand that what are the stages of the product development, and two stages are discussed in detail in this video, that is literature search, preformulation, and the DEC, that is drug excipient compatibility studies. So the upcoming videos will come, and in those videos the remaining part will be studied in detail. So stay tuned to this channel, Pharma Learning in Depth. Do like, share and subscribe to this channel, and thank you for watching the video.